hope you guys are rested, well rested. I hope you guys are okay. I hope you can use your 3D vision very well because we will need it a lot today to try to visualize how things are in embryo. Okay, we have finished with this picture. I edited it right now, you don't have it, but we have finished with this picture. You remember that this is a day 14 of the blastocyst stage. How do we call this together?
Conic plate.
Yes, we are going to call it chorionic plate. Chorionic plate is going to consist of 3 things, as you remember. The most outer is syncytial trophoblast. Then we have cytotrophoblast. Then which one is this? Pleuric mesoderm. Very good. What was the function of syncytial trophoblast? What did it do? On day 8, it started doing something. What?
The mother blood came into the lacuna.
Maternal blood vessels entered into the lacunas on day 11, 12. But on day 8, syncytial trophoblast starts to secrete a hormone. Which hormone is that?
HCG.
Very good. Human chorionic gonadotropin. What is the function of HCG?
It's the indicator in the pregnancy test.
Yeah, but that's not its function. It is an indicator, but it's not its function.
To constant.
Maintain
to replace
the
LH.
Yes. So to keep the corpus luteum alive. Do not forget that it doesn't bring LH back, right? LH will go down either way. So to maintain the corpus luteum alive, we need the HCG because HCG looks like the LH. What was the function of the corpus luteum?
Produces progesterone and estrogen.
Very good. It produces progesterone and estrogen. And you remember that progesterone is the one that keeps the endometrium thick. Now here we have the amniotic cavity. Here we have the secondary yolk sac. Why do we call it secondary yolk sac?
It's because the first one bursted.
Exactly. Why did the first one burst?
Because of the fluid. It didn't have any place, so it burst.
But what was the actual reason for it? What was the physical force that caused your primitive yolk sac to burst?
The extraembryonic cavity grew.
Okay, so the extraembryonic cavity, the chorionic cavity, as it was being formed, you remember, it was exerting pressure on the original yolk sac. The original yolk sac could not handle that much pressure, so it bursts, right? This is day 14. Is it correct to say that hypoblast is only located on the bilaminar germ disc?
No.
No, where else do we have the hypoblast?
In the yolk sac.
In the wall of the yolk sac. Very good. So within the wall of the yolk sac, we also have the cells of the hypoblast. What is connecting stalk going to become?
The future umbilical cord.
Very good. This is the future umbilical cord. What is exocoelomic cyst?
That is the primitive yolk sac. That was
the remnant of the primitive yolk sac, right? So it's the whatever little tiny part of it that sometimes is visible. So what we're going to do right now, we're going to talk about the early phase of placentation. Okay, so the early phase of making placenta. In your exam, they will say further development of the trophoblast. So you remember that syncytio and cyto, they are part of the trophoblast. So we are continuing with the topic, further development of the trophoblast, but basically what it means that we're going to start talking about placenta, early phases of the placentation. You remember that here we had the cavities. We call those cavities trophoblastic lacunas. You remember that on day 11, 12, this is when maternal blood vessels started to enter into these trophoblastic lacunas. So as a result, the maternal blood started to supply your embryo with nutrition. You need to remember that maternal and embryonic blood can never mix. Maternal and fetal blood can never mix. but maternal and baby's blood can occasionally mix. When? During delivery. When you're giving birth, there are lots of membranes rupturing, so occasionally you can see that mom's blood and baby's blood could mix a little bit. But in embryo, during pregnancy, we cannot allow that maternal and fetal blood are exposed to each other. It's not allowed. Why? Because we have two different immune systems. They will start to attack each other if the two bloods are exposed to one another. So keep that in mind. Keep that when maternal blood enters into the trophoblastic lacunas, it should never be exposed to the embryonic blood. Okay, they should never mix together. There should be a barrier between them. This is a very good drawing to show you, because how do we call this together? You remember that this is the chorionic plate. So this is this. It's just three of them without anything else. You remember that chorionic plate consists of the syncytial trophoblast, cytotrophoblast, and under it we have the extraembryonic somatopleuric mesoderm, right? So I'm talking about this area right here. It's exactly the same. Now, what is above it? What is at the top of the syncytial trophoblast? Of course, endometrium. So all of this should be your endometrium. It is not important for this semester. I highly doubt they will talk about it this semester. But for next semester, you need to remember that endometrium, which is pregnant, it has a different name. It is known as the decidua. Okay, so maybe they will ask this semester as well, because it is part of the placenta topic, but pregnant endometrium, it is actually known as the decidua. So what surrounds your blastocyst? Decidua, that's what surrounds it, right? So all of this endometric tissue, it is known as the decidua. So let's start making placenta. First of all, approximately on day 11-12, what was happening during day 11-12? Maternal blood vessels were entering, right? Simultaneously, you're going to see that syncytial trophoblast starts to penetrate deeper into the endometrium. It starts to grow some kind of columns, some kind of cellular columns, and these cellular columns start to protrude even deeper into the endometrium. Who wants to join the syncytial trophoblast? Of course, the underlying cytotrophoblast. So what do you see? On day 11-12, you start to see that primary villi appear. These primary villi are located in the endometrium. They're like fingers which are going into the endometrium like that, right? So this is the syncytial trophoblast. This is the endometrium right here. You see that syncytial starts to literally invade the endometrium forward, creating some kind of cellular columns, which we're going to call primary villi. So for the exam, how can they ask? They'll ask you, what does primary villus consist of, right? So what do primary villi consist of? The outer layer of the primary villus is, of course, syncytial trophoblast. It's this area right here. The inner layer is the cytotrophoblast. On day 14, you start to notice that somebody else wants to join the party, and this is the somatopleuric mesoderm. It wants to go into the core of the primary villus. Once somatopleuric mesoderm is in the core of the primary villus, you are no longer calling the structure primary, you're calling it secondary villus. So what is the major difference between primary and secondary villus?
The somatic mesoderm?
Exactly, the presence of the somatopleuric mesoderm. If it is in the core, you're going to call it secondary villus. If it is not in the core, you're going to call it primary villus. For the rest of your life, I want you to remember one thing. Whenever you see the word mesoderm, it's such a broad tissue. It can give rise to so many different things. Today, we're going to see that we have so many different mesoderms. But the two things that mesoderm always gives rise to is connective tissue and blood vessels. So blood vessels with blood, okay? Connective tissue and blood vessels, they always come from mesoderm. So for example, we have paraxial mesoderm. We'll learn it today. It gives rise to axial skeleton, but what else can it give rise to? To the connective tissue of the axial skeleton, to the blood vessels of the axial skeleton. This is extraembryonic somatopleuric mesoderm. It is mesoderm. So what is it going to do? It's going to differentiate to become blood vessels and connective tissue. Are we on the fetal side or on the maternal side? Of course, these structures are fetal structures, they're embryonic structures. So whose blood vessel is this? This is embryonic, this is fetal blood vessel. As soon as blood vessels start to appear here, of course, you cannot call it secondary villi. You must call them tertiary villi. So what is the difference between secondary and tertiary villus? Secondary villus still has the mesoderm, while the tertiary villus already converted that mesoderm to become blood vessels. So now, very important question for you guys. This is early placenta. In the late placenta, after 5th month of embryological development, which villi are you going to see?
Tertiary.
Tertiary.
And what is the keyword that you're forgetting?
Villus.
No, tertiary only.
Um,
You are not going to see primary. You are not going to see secondary. You're only going to see tertiary. Why? Why in the late placenta only tertiary villi will be present?
No mesoderm,
What is the core structure of the tertiary villus?
blood vessels,
Whose blood vessels?
fetal,
The
And
embryonic.
what is the purpose of placenta?
to give the fetal nutritions and oxygen.
Exactly. So if you're going to give the nutrition, where are you going to put it? You're going to put it in a fetal blood vessel, right? You're going to put it inside of the fetal blood vessel. So in the late placenta, is there really a need for primary or secondary villi? No, your embryo is growing. It needs more nutrition. So for sure, you're going to see that tertiary villi are going to be the only one. Okay, so the only ones. You can see the primary villi here on this picture. Do you see? So this is a primary villus. This is another primary villus. This is another, right? So they're starting to become secondary because you see that the mesoderm wants to join this area right here. So mesoderm is going into the core of the primary villus. So we can imagine that this is, for example, one tertiary villus. This is another tertiary villus right here. As you can see, between them, there is a space. The space is called intervillus space in late stages. But in early stages, what is this? What is another name for it in early stages? You know it.
Lacuna.
Very good. Lacuna, trophoblastic lacunas. Trophoblastic lacunas are these areas. So who is entering into this part? Maternal blood vessel. You're going to see that maternal blood vessel is going to enter here. So this pool will consist of the maternal blood. Tertiary villi will have fetal blood. What is between them? What is the barrier between maternal and fetal blood? Which layers?
Syncytiotrophoblastic and cytotrophoblast.
Very good. So do you see they don't mix? The maternal and fetal blood, they are never exposed to each other. Maternal blood will bring me oxygen, it will bring me nutrients. These things will just diffuse across the syncytio and cyto into fetal blood vessel, and through the umbilical cord, they will return to the embryo, right? In another intervillus space, you're gonna see we're gonna drop the toxins. We're going to drop the carbon dioxide this way. Maternal vein is going to be here. It will take all of those things and the mom will get rid of it. But this is late placenta. We talk about late placenta next semester. Early phase of the placentation, is it clear?
Do we need to know about HIV of maternal blood or RH of
Not
embryonic
really.
blood?
I don't know, nothing. Next semester we talk details placenta because this is just the beginning. Okay, Sabina, you had a question.
Yes, in tertiary villus, do you have mesoderm? It means that there is no mesoderm, there are just blood vessels because mesoderm became it, no?
But it didn't just become blood vessel, it also became connective tissue because that's what we're talking about, right? So this mesoderm that is in the secondary villus, it differentiated, it became fetal blood vessels surrounded by embryonic connective tissue. So what are these fetal blood vessels surrounded by? By mesenchyme, right? So in the tertiary villus, you already see that there is mesenchyme with the fetal blood vessels. In the secondary villus, you don't see that. You just see the undifferentiated mesoderm.
Okay, understand.
Sorry, I have a question.
Yeah.
So the space between two tertiary villas is intervillus space?
Yes, in the late placenta, but
yeah,
in
so
the early placenta,
it's the trophoblastic lacuna.
for sure. Yeah.
So then, so are they secondary villi or still tertiary villi?
But you're confusing. They are secondary villi or tertiary villi, either way. They aren't related to the space right here.
Okay.
It has no relation to them, right?
Mm-hmm.
I wrote here intervillus space because that's what the book says, but the book has a bit of a mistake, okay? Because we call this space between primary or secondary or tertiary villi. It doesn't matter which villi you have. Just the space between them in the late placenta is called intervillus space. But in the early placenta, which is our topic for this semester, it's not called intervillus space, it is called trophoblastic lacuna. So the villi don't matter. Just the space between them is called trophoblastic lacuna. Yeah?
Thank you.
Mm-hmm. So that's your early phase of placentation. Very important for the next semester, I want you to remember, placenta is not fetal nor maternal organ. It's both. Placenta is the organ which is shared by mom and by the embryo. You can't choose the option that says that it's only fetal organ, it's only maternal organ. Placenta consists of two parts. It consists of the fetal part and of the maternal part. So it's an organ that is both fetal and maternal. It's for next semester. Okay, we start the most horrible thing ever, which is called gastrulation. Okay, you're gonna see so many pictures from here, so many questions, so let's take a look. Basically, you remember that we have a bilaminar germ disc on day 14. This bilaminar germ disc consists of the top epiblast and bottom hypoblast. But we also have trilaminar germ disc, and trilaminar germ disc consists of the ectoderm, mesoderm, and endoderm. Gastrulation is the process of making trilaminar germ disc. That's what it means. Gastrulation, it means that we have to convert our bilaminar germ disc to trilaminar germ disc. We have to convert our epiblast, hypoblast to ectoderm, mesoderm, endoderm. Gastrulation begins during the 3rd week. Okay, so starting after day 14 until day 21, okay, so sometime there, we're gonna see that this is when we're gonna be creating the trilaminar germ disc. This picture is very important. They will bring this picture or a version of it that looks like this. Okay, so that looks like this. So either this picture will be brought up or this one. So let's try to understand this picture right here. You can see that the two cavities were cut off. the top cavity, amniotic, we can see only the wall here. It was fully cut off, right? And the bottom cavity, the yolk sac, we can only see the wall here. But you can imagine that amniotic cavity used to be here like this, and yolk sac used to be at the bottom. What do we see? We see the bilaminar germ disc, the top layer being epiblast and the bottom layer being hypoblast. Gastrulation begins with the formation of a structure which is called primitive streak. So this is the beginning of gastrulation. You can't start gastrulation until this structure is formed. Could you tell me on which layer primitive streak is formed? On epiblast or on hypoblast?
Epiblast.
On the surface of the epiblast. Sorry, on the surface of the epiblast, okay? On which floor cavity is it formed? At the floor of the amniotic or at the floor of the yolk sac?
Amniotic.
Amniotic, of course. So you can imagine, right? Amniotic is the top, yolk sac is the bottom. Primitive streak is formed at the floor of the amniotic cavity on the surface of the epiblast. The most important thing about primitive streak is that it is formed at the caudal end of an embryo. Okay, this word is very important in embryology, caudal. Caudal is the opposite of cephalic or cranial. It's the opposite. Cephalic or cranial end of the embryo is the head. Caudal end of the embryo is the tail. Your tail is your buttocks, your coccygeal region. Yeah, that's your tail. That's the area where we should be having our tail because we're mammals, right? So is it clear? Primitive streak is at the level or at the end of the caudal part of an embryo. How does the book let you know that this is the caudal end of the embryo? As you can see on the opposite, it shows you that there is a membrane. It's called oropharyngeal membrane. Oro means mouth. If this end is the mouth, this must be the opposite end. Okay, so this must be the caudal end right here. So I repeat, gastrulation is making trilaminar germ disc. It starts during 3rd week, and it begins with the formation of primitive streak on the surface of the epiblast, right, at the caudal end of an embryo. We can see exactly the same picture here. It's just a bit different view. Do you see? This view is more from the superior point of view. This is the cranial end of the embryo. This is the caudal end of the embryo. This structure right here is your primitive streak, right? This is primitive streak right here. I want you to notice that the cranial end of the embryo is much broader. Okay, this will be asked. And the caudal end of the embryo is much more narrow, right? So your tail is a bit more narrow. If you do not understand what streak means, it means groove. Groove means invagination. Do you see that on the surface of the epiblast, we're creating some kind of deepening. This deepening is known as the streak. A question is going to arise about this structure. Primitive node is located right here. So now I'm going to say a sentence. Listen to it because I can't repeat it twice. It's such a long sentence. And tell me if it is true. Primitive node is located at the cephalic end of the primitive streak, which in turn is located at the cephalic end of an embryo.
False.
Which part is false? The first half of a sentence or the second?
Second part. First is true.
Very good. The first half is true. Primitive node is located at the cephalic end of the primitive streak, for sure. This end is the primitive, this end is the cranial end, right? For sure, because it's closer to the head. But then it said that primitive streak is located at the cranial end of an embryo, which is not true, because primitive streak is located at the caudal end of the embryo. So I repeat the sentence in the correct form. Okay, I repeat it in the correct form, how it should be. Primitive node is located at the cephalic end of the primitive streak, which in turn is located at the caudal end of an embryo. Okay, that sentence is true. Yeah, that sentence is true. Okay, let's do gastrulation. Okay, let's do it. We have to draw it because it's a very easy process. We do a cross section through the primitive streak. We want to cut it through primitive streak exactly as it shows on this picture. I just want to draw it for you. Remind me, which cavity do we have at the top?
Amniotic cavity.
This is the amniotic cavity. Which cavity do we have at the bottom?
The yolk sac.
This is the definitive yolk sac right here. Which layer do we have here?
Epiblast.
Very good. So this is the epiblast right here. Which one do we have here?
Hypoblast.
Hypoblast. Is hypoblast only here?
No.
No, don't forget, I didn't draw it nicely, but don't forget we also have hypoblast this way, right? It's in the wall of the yolk sac as well. So what is this?
Primitive streak.
The streak.
Primitive streak. So this is the invagination on the surface of the epiblast, which we call primitive streak. So let's begin the gastrulation. Gastrulation begins when cells of the epiblast detach, migrate to the primitive streak, come through it, come to the underlying hypoblast, and keyword, displace it. They move it away. The incoming cells of the epiblast, they detach from it. They go through the primitive streak and start to literally swipe all of these cells of the hypoblast away. Where do they swipe the hypoblast? To the mesoderm on the top of the yolk sac, which is the splanchno-pleuric mesoderm. Okay, all of the hypoblast, which is located here and in the wall of the yolk sac, is being pushed to the splanchno-pleuric mesoderm. So would you find cells of the hypoblast in the bilaminar germ disc? Not anymore. Would you find cells of the hypoblast in the wall of the yolk sac? Not anymore. Where is this hypoblast? It's outside. It's in the splanchno-pleuric mesoderm. So now you have two empty layers, supposedly. They're empty. So who is going to occupy these layers? The incoming cells of the epiblast. They come in and fully occupy this area. They take over this new layer right here that is now empty. How do we call this layer? We call it endoderm. Endoderm arises, I repeat, when cells of the epiblast detach, migrate through the primitive streak, come to the underlying hypoblast and displace it. This word is crucial for next semester. You have to say it. If you don't say it, like, it's not good, okay? They displace the hypoblast. Where do they put the hypoblast? Outside, to the splanchno-pleuric mesoderm. The cells of the epiblast now take over this layer. As they take over this layer, now they're called endoderm. So now a very important question. In the wall of the yolk sac, do I have hypoblast or endoderm?
Endoderm.
Endoderm, very good. You will not find hypoblast anywhere except in the splanchno-pleuric mesoderm. So now endoderm is in this layer and in the wall of the yolk sac. Is that clear? A question for you. Just a sec. A question for you. Hypoblast gave rise to endoderm, correct?
False.
It's a false statement because it was displaced. It didn't do anything for the endoderm. Who gave rise to endoderm?
Epiblast.
Epiblast. Is that clear? Where is the hypoblast now? Splanchnopleuric mesoderm. It's outside, it's in the splanchnopleuric mesoderm. We continue. Cells of the epiblast keep on detaching, keep on migrating through the primitive streak, but they don't go to the endoderm, they don't. Instead, they start to occupy the middle area. They start to accumulate in the middle right here. These cells are known as the mesoderm. And the cells of the epiblast that didn't detach, they stayed where they are, right there on the surface, nothing, no movement whatsoever. These cells are becoming ectoderm. So now a very important question. Trilaminar germ disc arises from bilaminar germ disc. Is that sentence correct?
Yes.
How?
It's a false statement. Why is it a false statement?
But the cell detached.
The
Because it only
hypoblast.
comes
Because
from the epiblast.
hypoblast is not involved.
Exactly, because the epiblast is the only one that gives rise to it. Hypoblast gives you nothing in the trilaminar germ disc. It gives you absolutely nothing. So this statement is incorrect. It is on the gray area, and anatomy department has lots of gray questions, okay, that it's not technically correct, but still this is a false statement. Trilaminar germ disc arises from the epiblast of the bilaminar germ disc. It doesn't come from the bilaminar germ disc itself. Because if you say that, you're implying that hypoblast should give rise to something, but it doesn't give rise to anything in the trilaminar germ disc. Okay, you can see here very important FGF8, E-cadherin, brachyury expression. First of all, the first thing that you should be asking me, why the hell are they detaching? How are they even how is it possible that cells that are sitting in the layers randomly start to detach and go migrate some kind of places? It's the fault of this one. Fibroblast growth factor 8. In embryo, we're going to see lots of FGF, especially next semester, okay? Fibroblast growth factor. There are many of them. The one which is important for you right now is the FGF8. Who secretes fibroblast growth factor 8? Cells of the epiblast near the primitive streak. So those cells of the epiblast, which are adjacent to the primitive streak, they're actively secreting fibroblast growth factor 8. This fibroblast growth factor 8 has two functions. Number one, it inhibits E-cadherin. Could you remind me what is E-cadherin?
Zecom.
What is E-cadherin? This is our first histo self-control test. We saw it there.
For the dark junction between the cells.
It's
The
the
lateral side.
Exactly. It's the part of the zanula adherens and macula adherens. Hopefully you remember. It's the part of the junctions that hold cells together. E-cadherin is the glue that holds cells together. So we're answering the question, how are the cells even detaching? They're capable of detaching because E-cadherin, the thing that holds them together, is not there. Who inhibits E-cadherin? FGF8. Who secretes FGF8? The cells of the epiblast, which are near the primitive streak, okay? That was the first function of FGF8. The second function of FGF8 is that it is an attractant, okay? It's a chemoattractant. There's lots of FGF8 near the primitive streak, and the detaching cells of the epiblast go ballistic for it, okay? So they really want to go near it. So it causes them to come to the primitive streak, right? So it attracts them to come this way specifically. Third function of FGF8 is that it stimulates brachyury expression. To simplify, what does this thing do? Try to answer this question. I am an epiblast. If I migrate first, what am I? If I migrate first, what am I destined to become?
Endoderm.
Endoderm. I am an epiblast. I don't migrate whatsoever. I stay where I am. I am
Ectoderm.
ectoderm. Agree with me, for mesoderm, it's weird. It migrates second. It migrates second. So how does it know? Should I migrate now and be with endoderm, or should I not migrate whatsoever and be with ectoderm? How does it know? It's like, you know, like when you're busy, you give a child something to play with until you do your thing, right? So you keep them, I don't know, here's a pen, draw me something, and I wash the dishes now. Exactly the same thing is happening there. The cells of the epiblast, which are destined to become mesoderm, are busy expressing brachyury. By the time they're done expressing brachyury, endoderm is already formed. So now I can detach, migrate, and give rise to the mesoderm. Do you get it? Brachyury expression is only for the cells of the epiblast, which are destined to become mesoderm, because they migrate second. It's weird. If I migrate first, it's clear as day. I migrate first. If I don't migrate, it's also clear. But for mesoderm, it's a bit confusing. Do I migrate or do I not migrate? What do I do? Give them brachyury. While they're expressing it, endoderm is formed, and then they will migrate and become mesoderm. Okay, this very important. They keep saying, no, we aren't going to ask this every year. It's the same thing. We're not going to ask it, and then every year it's the past paper question you see, and it's there. Okay, how do you know that this is the head? On this picture alone, how do you know?
It's wider.
Very good. It's much broader, it's much wider. How do you know that this is the tail? Because it's narrower. But how do you know that this is the head? Like, it's not enough for the cells to be like, is it broader? Yeah, it's the head. It's not enough for cells. We need some kind of growth factors. We need some kind of transcription factors to be aware that this is actually the head and this is actually the tail. Randomly, before gastrulation, randomly, so approximately on day 13, 14, randomly before gastrulation, 5 genes are being expressed. I highly doubt you need to know them, but next semester you definitely need to know them. Definitely. This semester I highly doubt. Next semester definitely you have to. These genes are called OTX2, LIME1, HESX1, Cerberus, and Lefty. These 5 genes, the area where they are expressed, are forcing that part of the embryo to become head. So randomly, these 5 genes choose a side, either one end of the embryo or other. If I choose that end, that's the head. If I choose the other hand, that's the head. Okay, so randomly this process happens. So you have a disc, you have an embryo as a disc, and randomly during day 13, 14, we choose this is going to be the head or this is going to be the head. Okay, how do I know that this is the head? Because of these 5 genes. The area where these 5 genes are expressed is known as AVE. So this part of the embryo where these genes are located is known as AVE. AVE means anterior visceral endoderm. So anterior visceral endoderm is just the part where these 5 genes are going to be located. What is at the caudal end of the embryo? The primitive streak with the primitive node. So how do we know that it's going to be at the caudal end? Because the head is occupied. OTX2, LIME1, and HESX1 are inhibiting formation of primitive streak in the head. Even if you try to make S in the head, you won't. You won't because of this OTX2, LIME1, HESX1. So head is at one end, and it stimulates formation of primitive streak at the opposite end, at the caudal end. To make sure that this is definitely the tail, primitive node starts to secrete something which is called nodal. Okay, it starts to not secrete, sorry, it's my fault. It doesn't secrete it, it expresses it, is a transcription factor. Okay, it expresses nodal. And nodal definitely pinpoints that this is the tail. So if I were to ask you which 5 genes are for the head, you're gonna name these ones. If I ask you which gene is for the tail, you're gonna say nodal. Nodal is the one that tells you this is the caudal end. But another very important process in embryo is called left-right sideness or laterality pathway. How do we know that this is the left side? How do we know that this is the right side? That's the idea, right? So how do we know? Notice that nodal is being expressed at the tail, but not only at the tail, it's also being expressed at the left side of the tail. Nodal is always on the left. You remember FGF8? The cells of the epiblast around primitive streak were secreting it. Take a look, it's also being secreted more to the left. Majority of it accumulates on the left side. Together, nodal and FGF8 upregulate or induce expression of LEFT2 and PITX2. And if I ask you which 2 transcription factors are responsible for the left side, you're gonna name these ones. You're gonna name LEFT2 and PITX2. Please, for the love of God, do not choose lefty and lefty2. They will give you the option. They will say which 2 transcription factors are responsible for the left side of the embryo. Don't choose lefty and lefty2. What is lefty responsible for?
The caudal.
This is the
Right side.
caudal end of the embryo. What is lefty responsible for?
Cranial end.
For the head formation, for the cranial end formation. Lefty is responsible for making the head. Lefty2 and PITX2 are responsible for making the left side. If this is the left side, automatically the other one is right. So this is why they do not know many transcription factors that are responsible for the right side. The only one that they know is snail. Snail is the only transcription factor which is responsible for the right side. So again, which transcription factors or which genes are for the head? These 5. Which ones are for the left side? Lefty2, PITX2. Which one is for the right side? Snail. Is that clear? Sonic hedgehog is a very important structure in embryo. It regulates so many processes. It controls so many processes. And one of the processes that it controls, it makes sure that none of the left side transcription factors are expressed on the right. Okay, so sonic hedgehog will inhibit lefty2 and PITX2 on the right side. If it doesn't, if for some reason lefty2, PITX2 end up on the right, you could have 2 disorders, dextrocardia or situs inversus. Dextrocardia is the mild form. It basically means that your heart is not shifted to the left, it's shifted to the right. That's it. Situs inversus is so interesting, okay, so interesting. It's basically a mirror image of your organs. So heart is on the right, liver is on the left, spleen is on the right, right? So every single organ is in the opposite way as it should be, exactly mirror image. That's situs inversus. Is it deadly? No, those people are fine. It's just that for surgeons, they will have to like switch everything when they're operating on them because it's completely the opposite way, right? So where is going to be inferior vena cava? On the left. Where is aorta? On the right. It's crazy, okay? Everything is the opposite way. So that's situs inversus. So as you can see, sonic hedgehog is crucial for that, right? So it makes sure that definitely everything is kept in order. These pictures are very important. Sure, they will come up as well. So here we can see two views of our embryo. One of them is a cross section. One of them is a sagittal section. Can somebody show me on themselves how is sagittal section? I can see Sabina is showing coronal. That's coronal section like this. Okay, I think Gilly was showing. She was showing something like this. Okay, so yeah. Okay, so sagittal is this. It's this. It's like that. But if I say mid sagittal, right? It's this. It's exactly in the middle. It's exactly right like that. So there are two pictures here. One of them is mid sagittal section of the embryo. One of them is a cross section, transverse, right? So cross or transverse. What do you think? Which one is this? This top one. Mid sagittal or transverse?
Mid sagittal.
For sure, mid sagittal. How do I know? Because that's the head right here. That's the tail right here. I can actually see head and tail. So mid sagittal section. Okay, I can clearly see it. How do you know that this is the cross section? Because you can't see head and tail. You cannot see the ends of the embryo. You can just see like a transverse section. So take a look. This is the head of the embryo. This is the tail. This cavity is the amniotic cavity. This is the yolk sac right here, right? We have three germ layers, the top being ectoderm, middle being mesoderm, and the yellow being endoderm. You can see two membranes. You see the cranial membrane called oropharyngeal membrane, and the caudal, the tail membrane called cloacal membrane. Notice that oropharyngeal and cloacal membrane consist of tightly adherent ectoderm and endoderm. Blue is ectoderm, yellow is endoderm, orange is mesoderm. Do you see any mesoderm in them? No. Okay, so tightly adherent ectoderm and tightly adherent endoderm together. What do? They cover the entrance and exit of the digestive tract, the future digestive tract. In embryo, primitive oral cavity, so the mouth itself, right? So oral cavity, primitive oral cavity is called stomadeum. Okay, we're gonna call it stomadeum. It's a very unfair question, but I see it in past paper. Okay, next semester we'll talk about it more, but for now, just learn this word, stomadeum. Stomadeum is where the teeth are, okay, the future oral cavity. How do you call the anal doctor? Proctologist. So how do you call the primitive anal cavity?
Proctodeum.
Very good, we call proctodeum. Okay, so stomadeum is the future oral cavity. Anal cavity is known as the proctodeum. So if I draw it very weirdly, that's the mouth right here. Okay, so that's the mouth, this is the tongue. And on the opposite end, of course, I don't draw the whole body. On the opposite end, that's where the anal cavity is. So the mouth is stomadeum, the anal cavity is proctodeum. So where are these membranes? They're right here. Oropharyngeal membrane will be here. Cloacal membrane will be here. So if I ask you a question, oropharyngeal membrane ruptures into what? Where does it rupture into? Stomadeum. Where is the oropharyngeal membrane in you? You don't have it, but it used to be. If I open my mouth, you know, there is uvula hanging. Do you know? If I open my mouth, the uvula is hanging there. Please don't make me open my mouth. Okay, so uvula this way. That's where oropharyngeal membrane is. So it will be rupturing into your mouth. Okay, the same way for the opposite end. Okay, so it will be where the anal cavity is, just rupturing there. So that's the questions in the self-control test. They ask you, where do these membranes rupture into? Okay, oropharyngeal membrane will be rupturing into the stomadeum. Cloacal membrane will be rupturing into the proctodeum. Anybody knows what is cloaca? Anybody is a villager? It's right here. Yours truly. I grew up with chickens, cows, horses. Okay, what is cloaca?
Cow's poo.
Nope. Nope. Cloaca is a, okay, imagine a vagina and anus together. Birds pee, poop, and give birth from the same hole, and that hole is called cloaca. It's like a common orifice, it's like a common hole for things. So fun fact, embryos have cloaca. Okay, we're gonna learn how to separate this cloaca next semester, but embryo, they have a common orifice for everything, and that is called cloaca. So cloacal membrane is technically going to be rupturing into cloaca, but we're gonna say this sentence next semester. This semester, you're saying that cloacal membrane is rupturing into proctodeum, into the future anal cavity. But I repeat, for the next semester, you are not going to see proctodeum immediately. At first, there's going to be cloaca, the common orifice from which embryo is pooping and peeing. Okay, but don't forget, embryos don't poop. They pee, but they don't poop. So yeah. Okay, here we have the ectoderm, here we have the endoderm. There is a canal between amniotic cavity and the yolk sac, and this canal is called neuroenteric canal. It is closing 2 to 3 days. Okay, so this is approximately day 21, day 24 to day 27. This is when this canal will close. So you're going to have an intact ectoderm, intact mesoderm, intact endoderm. You won't have any communication between the amniotic cavity and the yolk sac. Behind the oropharyngeal membrane, we have the precordial mesoderm. Precordial mesoderm will give rise to your forebrain. Okay, the forebrain. Forebrain are your cerebral hemispheres. Okay, so as you can see in embryo, brain is behind the mouth. Like this. Okay, so this is the mouth, the brain is behind. Why is it called precordial mesoderm? Because it is anterior to some kind of cord. How do we call this cord? We call it notochord. This should be already ringing a bell. How do we call a remnant of the notochord in the adult body?
Spinal cord?
Belly button?
Histo. Remnant of the notochord in the adult body. Okay, I'll start talking and you'll remember. So notice that notochord is not everywhere. Do you see? It's only in the middle of the embryo. So if I show it with my hand, right, this is the head, this is the tail. Notochord is literally going to be, I don't have a pen. It's literally going to be here. It's going to be just in the middle. So every time you do a cross section, you will always see notochord as a dot, always, because every time cross section, notochord is a dot. But you can only see notochord as a line if it is a mid-sagittal section, right? So you can see I'm touching my pen, so it is a mid-sagittal section right here. Notochord is mesodermal origin. It acts as a basis for the axial skeleton. Okay, so it saves a spot for the future axial skeleton to arise. And it disappears, mostly, but it leaves a little remnant somewhere in the axial skeleton. Anybody remembered?
In the vertebral column.
Where exactly?
We call nucleus pulposus.
Nucleus pulposus, very good. In the intervertebral disc, we have two parts. We have the jelly center, and we have the fibrocartilage, which is in the periphery. How do we call the periphery of the intervertebral disc?
Annulus fibrosus.
Very good. We call it annulus fibrosus. This is the fibrocartilage. In the center, we have the nucleus pulposus. Nucleus pulposus is this hyaluronic jelly, which is the remnant of the notochord. So notochord will be disappearing. The center, I repeat, it kind of gives rise to, it kind of shows you where the axial skeleton will be. Okay, so that's what it does. So when you do a cross section of the embryo, you know that right there in the middle, right? So you're gonna have the axial skeleton for sure. Last thing on this picture, we have here something called allantois. Allantois is an outgrowth of the posterior wall of the yolk sac. So which tissue does allantois consist of? It consists of the endoderm, right? Because we have endoderm in the wall of the yolk sac as well. You may not see the word outgrowth. You may see the word diverticulum. Diverticulum is just a medical name for outgrowth. Okay, so like your appendix is diverticulum. Okay, it's just an outgrowth of some sort. What is the function of allantois? Here I wrote you that there is no function because according to the embryo 1, there is no function, but according to embryo 2, next semester, there is. Allantois will be participating in creating your bladder. It's very needed to make bladder. Okay, so you can see that for now, allantois is actually in your umbilical cord. So for a very, very short period of time, the bladder of the embryo is in the umbilical cord. But we're still very early. This is day 21. Okay, so at the later stages, for sure, it's going to be in the body. So allantois is a diverticulum of the posterior wall of the yolk sac, and it participates in making bladder. Okay, this picture, is it mid-sagittal or cross?
Cross.
How do you know?
Notochord is dot.
Very good, very, very, very good. Okay, so this is the proper answer, especially next semester. If they draw it for you, you should always look for the notochord. I repeat, I show it with my hand. Do you see it's broader here and it's much more narrower here, right? My hand, broader here, narrow here. So you know that this is the head of the embryo. This is the tail of the embryo. Notochord will always be like this. It will always be right there in the middle of your embryo. I repeat, this is the head, this is the tail. So notochord is here. So if I put it like this right now, right? So my head of the embryo is here, my tail of the embryo is here, notochord is a line. So remember that every time you do a cross section of the embryo, notochord will always be a dot, always little tiny dot in the center. Every time you do a mid-sagittal section, you will see a notochord as a line. But what if I do sagittal section? Will I see notochord sagittal? No, because it's too far away, right? So notochord's in the middle. Only in the mid-sagittal section, you will see the notochord. So you know that this is a transverse section because notochord is a dot. So again, top cavity, amniotic cavity, ectoderm, notochord. We have two mesoderms on the sides of the notochord, and we have endoderm in yellow. Notice that endoderm is also in the wall of the yolk sac as well. Remind me, which tissue is on the top of the amniotic cavity? It's going to be extraembryonic, somatopleuric, mesoderm. Which tissue is on the top of the yolk sac?
Extra
Pleuric.
Pleuric mesoderm. Okay, so splanchnopleuroic mesoderm. Okay, guys, 2 minutes break. I'm just gonna go to the bathroom and we'll continue, yeah? So let me continue. So, as I have said, on this picture, we have the ectoderm at the top, notochord in the middle, endoderm at the bottom. And notice that notochord sort of divides your mesoderm into two halves, right? So do you see that there's half of the mesoderm here, and there is a half of the mesoderm here. This mesoderm is intra-embryonic mesoderm, because where is your embryo? It's your trilaminar germ disc. The mesoderm on the top of the amniotic cavity and yolk sac, this is going to be the extra embryonic mesoderm because it is outside of the embryo. Because notochord is in the center, we have said that it divided your mesoderm into two. The further differentiation of the intra-embryonic mesoderm, that it starts to divide into three pairs. That part of the mesoderm, which is closer to the notochord, this first pair, is known as the paraxial mesoderm. And you can guess that paraxial mesoderm is going to give rise to the axial skeleton, right? We said that notochord, it simply pretends to be axial skeleton, while paraxial mesoderm is the one which will definitely give rise to it. Right next to the paraxial, so this area right here, we're going to have intermediate mesoderm. Intermediate mesoderm creates urogenital system. Your entire urinary system, except for the bladder, and genital system, doesn't matter female or male, will come from intermediate mesoderm. That's the only thing we say this semester. Intermediate mesoderm gives rise to urogenital system. That's it. Next semester, we'll talk about how and what and where and etc. Right next to the intermediate mesoderm, we're going to have lateral plate mesoderm. But I want you to notice that the shape of the lateral plate mesoderm is V. Do you see it? This is the V shape of the lateral plate mesoderm. So again, I point, you tell me, which one is this?
Paraxial.
Paraxial, right next to it.
Intermediate.
Intermediate, and the V-shaped one is?
Lateral plate.
Lateral plate. Notice that the V is very interesting because part of the lateral plate is under the ectoderm. Part of the lateral plate is above the endoderm. Remind me which tissue is here? The extra-embryonic somatopleuric mesoderm. How are you going to call this part of the V? What do you think?
Somatopleural mesoderm.
But extra or intra?
Extra.
But extra is outside of the embryo. Lateral plate is in the embryo. We said that this V shape is in the embryo. So this little tiny part of the V under the ectoderm is going to be intra-embryonic somatopleuric mesoderm. Another name for the intra-embryonic somatopleuric mesoderm is also known as the parietal mesoderm, and the book got it wrong. Okay, it's not this, it's this little tiny part right here. This is the parietal mesoderm. What is lining the yolk sac? As you remember, extra-embryonic splanchnopleuroic mesoderm. So what do you think? What is this part of the V will be? Intra-embryonic splanchnopleuroic, another name for it, visceral mesoderm. Let me draw it for you so you get it. Okay, so where can I draw? I cannot draw anywhere. Okay, draw here. So we said that we have I draw just one part, yeah? Just one part. Here we have which cavity at the top?
Amniotic.
Amniotic. Here we're gonna have the yolk sac. Which layer is here like this?
Ectoderm.
ectoderm,
This is the ectoderm. Which layer is here?
endoderm,
Endoderm. Which mesoderm would be here on the two sides?
Lateral, please.
Lateral plate. What is the shape of the lateral plate?
V shape,
V. So just draw a V. This is one part of the V. This is another part of the V. Which one is right next to it here?
The
intermediate,
Intermediate. Which one is right next to it?
paraxial.
paraxial,
And then we have
notochord,
notochord. So do you get where we are, right? So at the top, we have the ectoderm. At the bottom, we have the endoderm. We're just looking at this lateral plate, at this V. Which one is at the top of the amniotic cavity here, coming down like this?
somatopleuric,
Extra embryonic somatopleuric mesoderm, because it is outside of the amniotic cavity. Which one is here outside of the yolk sac?
extraembryonic splanchnopleuric,
Splanchnopleuric mesoderm. Do you see there is a part where they're continuing with one another, right? Where somato starts to kind of continue with this part and splanchno starts to continue with this part. But this is the border. Everything else, which is to the left of this, is extra. Everything else to the right of this is intraembryonic, because this is your trilaminar germ disc right here. So as a result, we just said the lateral plate is the V shape. So that part of the V, which is closer to the splanchno, will be called intraembryonic splanchnopleuric mesoderm. Another name for this one will be known as the visceral mesoderm. This part of the V, which is closer to the somato one, do you see they're continuing with one another? You're going to call it intraembryonic somatopleuric mesoderm, another name parietal mesoderm. So quickly, under ectoderm, which layer do we have?
parietal mesoderm,
Parietal mesoderm. Parietal mesoderm is under the ectoderm. We're talking about lateral plate, yeah? I'm just in relation to lateral plate. So under the ectoderm, which part of the lateral plate do we have? The parietal mesoderm. Above the endoderm, we have the
visceral,
visceral. Yes. Last two names. We have something which is called somatopleur. And we have something called splanchnopleur. You again the same way, yeah. So somato and splanchnopleur. What is somatopleur? It's this. So could you tell me what is somatopleur?
ectoderm,
And the parietal.
And parietal mesoderm of the lateral plate. So ectoderm and parietal mesoderm of the lateral plate are called somatopleur. What do you think splanchno is?
The visceral and the
endoderm. Yeah. Okay, so visceral mesoderm and the endoderm. That's going to be the splanchnopleur. Are those terms clear?
Can you repeat, please, what makes the somatopleural?
Somatopleur is this area. This area is your ectoderm and parietal mesoderm. So that part. That means that splanchnopleur is the visceral mesoderm together with endoderm.
Okay.
Mm-hmm. Parietal mesoderm is continuous with the extraembryonic somatopleuric. Visceral mesoderm is continuous with extraembryonic splanchnopleuric. Is this clear? Okay. Derivatives of the ectoderm. So now what is left for us to do is just talk to ourselves. What can ectoderm give us? What does endoderm give us? What does paraxial give us? Intermediate and lateral plate. Okay, so beginning of the third week, we talked about it, blah, 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 blah. Okay, let's go to this picture. Basically, the appearance of the notochord induces overlying ectoderm to thicken. Okay, do you see that on the top of the notochord we have a little bit of the ectoderm, and this ectoderm thickens. So I give you the borders from here until here. So this area right here will be thickening, it will become stratified because of the notochord. This part of the ectoderm has a name. It is known as the neural plate. You can guess that neural plate will become something neural, right? So it should give rise to something of the neural tissue. Will this tissue be neural? Will this ectoderm be neural? Why not?
Because it's not in the space.
Because it's not thickened.
Because it's not thickened. And why is it not thickened? Because there is no
Notochord.
notochord, because notochord is not there. Okay, so this will be your surface ectoderm. So is it clear? So this part of the ectoderm is surface ectoderm. This is your neural plate. And then this is another surface ectoderm. What do we mean by surface ectoderm? General things that ectoderm give you. Ectoderm gives you things in your body that are needed to communicate with the outside world. So that's what ectoderm gives you. Ectoderm is always outside, okay? It's always the part of your embryo which will help you to communicate with the outside world, will help embryo to communicate with the outside world. Could you tell me what is the one thing which is facing outside?
Skin.
Say better.
The
Serum.
Skin has 3 layers. I taught you this in histo.
That's some cognition.
Epidermis.
Epidermis. Okay, there's epidermis, dermis, and hypodermis. So the one which is facing outside is actually epidermis, the stratified squamous keratinized epithelium. So when I say surface ectoderm, that's what I mean. I mean epidermis. Every time you hear me say surface ectoderm, I'm talking about epidermis. Okay, I'm talking about that part because dermis will come from somewhere else. Now, what about this neural plate? Well, think about it. We just said that ectoderm is needed to communicate with the outside world, right? Do I need a brain for that? Hopefully, okay, so hopefully I do need a brain to communicate with the outside world. So the entire central nervous system, which consists of the brain and spinal cord, should also come from the ectoderm. And where is it going to come from? Well, from the neural plate. Okay, so neural plate is that tissue which will give rise to the spinal cord and brain. So how do we talk about it? So how do we call a process of creating spinal cord and brain? This process is called neurulation. Is this picture clear? So I don't have to go back to the previous one. Do you see this is the neural plate, right? So do you see it does this? It sort of starts to fold. Okay, I'll just talk about it on this picture, never mind. Okay, so this is the ectoderm right here. This is the surface ectoderm, your future epidermis, the same as here. This is also surface ectoderm, your future epidermis. In the center, we have the thickened stratified ectoderm, which we're gonna call neural plate. Neural plate will start to invaginate. You're gonna see that there's some kind of deepening present here. This deepening is called neural groove. Neural groove will have 2 elevated components right here. These elevations are called neural folds. If I am trying to create a tube, what should neural folds do? Of course, you can understand, they should fuse together. When they fuse together, a structure arises, which is called neural tube. Which kind of neural tube do you have? This is your spinal cord. Okay, so spinal cord and brain, they are part of the neural tube. But notice that neural tube should not only be made, it should also detach from the rest of the surface ectoderm. Okay, it should detach from the rest of the surface ectoderm as on this picture. So if I show it on a piece of paper, this is your ectoderm, right? So this is the neural plate, surface ectoderm, surface ectoderm. You did something like this. How do we call this structure?
Neural
fold.
groove.
Neural groove. How do we call these elevated parts, this one?
Neural fold.
Neural folds. What do neural folds do? They do this. They fuse together. They come close together, and you have created neural tube. The problem is, do you see that my neural tube is still connected to the surface ectoderm? What I'm holding, this is surface ectoderm. So it's still connected to this. So what does the neural tube need to do? It needs to detach from the rest of the surface ectoderm. It needs to detach from your skin. You do not want your spinal cord to be connected to your skin. You don't even know anatomy, but you know spinal cord is in the vertebral column. It's inside of it. It's not even close to the skin whatsoever. So of course we should detach this neural tube. Is that clear?
Inside there is a lumen or amniotic fluid.
Of course, there's a lumen, yes, but we're going to fill that lumen with something. We will learn exactly 1 year from now in neuroembryology. What are we going to fill it with? Okay, but yes, this is a neural tube. How do they show you neural tube in your book? Like this? Okay, so that's the neural tube that you have created. Neural tube consists of the neuroectoderm. If I make the neural tube, right, like this, do you see it has the openings? I can actually see you from these openings right now. It has a hole. This is the anterior hole right here, and this is the posterior, right? So that's your neural tube. It has holes. I can see you through those holes. Those holes are called neuropores, those openings in the neural tube. There is anterior neuropore, which is closer to your head, and there is a posterior neuropore, which is closer to your tail. Do you want your brain to have a hole here? No. So we should close those neuropores. Anterior neuropore closes on day 25. Posterior neuropore closes on day 28. Once they do, that's it. You have your normal central nervous system, okay, that is consisting of the brain and the spinal cord. Is that clear? Okay, there are some problems with the neural tube. Sometimes neural tube does not detach from the ectoderm. It's connected to it, okay? Do you see here? It should be fully separated. Sometimes it stays connected. As a result, 2 disorders could arise: spina bifida and anencephaly. What is the difference between them? Location. Spina bifida, as you can see, spinal cord is exposed to the outside because it's still connected to your skin, right? So it didn't detach, it's still connected to your skin. So spina bifida is a neural tube defect, which is below cervical region. So it can be in your thoracic region, in your lumbar, in your sacral. Most commonly, it is in the lumbar region. Anencephaly means absence of brain. Does anybody know this baby? No? No?
It was a case a few years ago in the United States, if I'm not mistaken.
UK. It was in the UK. Yeah. So this baby is British. He died recently, by the way. He was born in 2014, and he died 2020. What do you think? How long can you live without a brain?
How did he even survive? I mean, you need.
That's why they call him the miracle baby. He's a miracle baby. He lived for 6 years. How long do you think the average anencephalic baby survives? Like maximum 3 months, maximum. Okay, so because you need a brain to live, you need a brain to function, you need the brain for everything. But they call him the miracle baby because he survived for like 6 years. Okay, so yeah. So as you can see,
how did, like, he function?
there is a documentary. I don't remember his name. to be honest with you, but if you Google anencephaly miracle baby, there was a documentary about him. They say how, but like his brain stem was intact, so as a result he could breathe, he could eat, and etc. But the brain part was gone fully. So anencephaly is that, okay? So if your neural tube does not detach, if it's still connected to the skin, as you can see, this baby has no calvaria. This is whatever brain he has, and with hairs on top of it, skin. So if you do this, you're touching the brain, basically. Okay, you're touching the brain. So anencephaly and spina bifida, I repeat, they're the defects of the neural tube. This is not important. I just want to get your attention to two very important things. Do you notice that ectoderm gives rise to two very important things? Your epidermis, skin, and brain. Agree with me that they aren't the same. Your brain and skin aren't the same. So how the hell one tissue is capable of giving rise to two things? Basically, the default state of ectoderm is to become neural tissue. If you look at the ectoderm, all of it is supposed to become neural tissue. All of it is supposed to be your nerves. Because we need noggin, chordin, and follistatin. Okay, these are the molecules which are making sure that your neural tissue becomes neural tissue. Anybody knows follistatin? You know, pregnant lady takes folic acid, but why do we take folic acid? To prevent anencephaly and spina bifida, to prevent neural tube defects, because it will create follistatin. And follistatin, noggin, and chordin are needed to create a normal neural tube, a normal central nervous system. But I still make the skin somehow. And the growth factor, which is responsible for the skin, is called bone morphogenetic protein 4, okay, BMP4. BMP4 is going to be typically secreted by the mesoderm, okay? So a part of the mesoderm, which is under the ectoderm, so this mesoderm right here, it will be secreting BMP4. If BMP4 is present, ectoderm will become skin. If BMP4 is not present, ectoderm will become neural tissue. So of course, it's natural that the noggin, chordin, and follistatin, they're inactivating BMP4. Okay, so in this little tiny part, even if there is BMP4, right, noggin, chordin, follistatin, they will inactivate that. So just know BMP4 and these three, and know what do they do with your ectoderm. That's the most important part. That's it. There is a special population of cells which are called neural crest cells. Neural crest cells arising from the neural fault. Okay, that's what they do. In America, neural crest cells are known as the fourth germinal layer. You know, we have three: ectoderm, mesoderm, endoderm. In America, there's a fourth, neural crest cells. They give rise to these things. Okay, so just all you have to do is just read them through, but typically they talk about Schwann cells. Okay, in the exam, you will see that they're saying Schwann cells arise from the neural crest cells. Correct? Correct. Okay, everybody knows what Schwann cells are? What do they do?
These are the cells that myelinate the nerves.
Exactly, and myelination is needed for faster action potential, right? So if I don't have it, I'm a vegetable, basically. So Schwann cells are very important, okay? So they're very important. Okay, we talked about that, lateral plate, okay? So we talked about it. Could you tell me, how do we call this?
Parietal.
Parietal mesoderm. How do we call this?
Visceral mesoderm.
Visceral mesoderm. So these are of the lateral plate. Paraxial mesoderm. Okay, paraxial mesoderm is the one which is in the center, surrounding your neural tube and notochord. Do you see there's a part of the paraxial mesoderm here and part of it is here? Agree with me that neural tube should be in the vertebral column, right? So majority of it is in the vertebral column. So paraxial mesoderm should give rise to the vertebral column and skull. Majority of the paraxial mesoderm becomes vertebral column. So you can imagine that it is not a brick of tissue. It is actually segmented. Paraxial mesoderm has little tiny units like on this picture. These are the units of the paraxial mesoderm. They're segmented. Why are they segmented? Because vertebrae. We have one vertebra here, one here, one here. So of course it has to be segmented. How do we call the segment of the paraxial mesoderm? How do we call the unit of the paraxial mesoderm? We call it somite. Okay, so that's the name for it. So these, they always point for you here. This is always a somite. So these are somites. When does the first pair of somites appear? Occipital region, day 20. From the day 20, every day, 3 new pairs of somites appear. So could you tell me on the 22nd day, how many somites are you going to have? How many pairs of somites are you going to have? Knowing that on day 20, we have first pair of somites, and then every day, 3 more pairs appear. So day 22, how many pairs would you have?
12.
7.
7. Why 7? Let's do day 20. First pair of somites appear. Day 21, how many more will appear? How many pairs will appear on day 21?
3.
3 pairs. Day 22, how many pairs will appear?
3.
3. So I can't draw it, but you can imagine that there are 3 more there. So count 3 plus 3 plus 1 is
7.
7 pairs. On day 22, you're going to have 7 pairs. On day 23, you're going to have
10.
10 pairs, because 7 plus 3. Every day you add 3 more pairs. You're done making pairs by the 5th week. You have made 42 to 44 pairs of somites. Out of those pairs, you're going to see that 4 are occipital, 8 are cervical, 12 thoracic, 5 lumbar, 5 sacral, 8 to 10 coccygeal. Does it look familiar? That's your future vertebrae. Yeah, so that's your future vertebrae right here. So a question for you guys. Tell me if it is correct. I can know the age of the embryo by simply counting somites.
True.
Absolutely, that's what we just did now. If I see 10 pairs of the somites, how old is that embryo?
That's it.
It's 23 days old. Yeah, it's 23 days old. If I see 13 pairs of somites, it's 24 days old. If I see only 1 pair of somites, it's 20 days old, right? So I can definitely know the age of the embryo by simply counting somites. Is this how many vertebrae you have? 42, 44? Definitely not. Okay, so definitely not. So you have made too many. Some of them need to disappear. Some of them need to fuse out. The first occipital pair of somites will disappear, and last 5, 7 of the coccygeal will disappear as well. Okay, so 5, 7 of the coccygeal, 1 of the occipital will disappear, and the remaining pairs, that's your future vertebrae. Okay, so that's your future vertebrae right there. Each of the somite, so this is just one somite here, has 3 clusters of cells, each of them. So if I draw one somite, instead of one somite, I can see myotome, I can see dermatome, and I can see sclerotome. What do you think myotome will give rise to? It will give rise to the skeletal muscle of the axial region, right? So skeletal muscle of the axial region will come from here. Sclerotome will give rise to the cartilage of the axial region, because your vertebrae arise through endochondral ossification. So first we make the cartilage in the shape of the vertebra, and then we convert that cartilage to become bone. What does dermatome give you? Don't say skin. Be more specific.
Dermis of the skin?
Epidermis.
Very good. Dermis. Where does epidermis come from?
Surface.
Ectoderm. It comes from the surface ectoderm. Dermatome gives rise to dermis of the skin. And what is so special of the dermis? You don't know that, but it's actually possessing nerves. Okay, it possesses nerves. Homework for you is to Google dermatome. Just Google anatomy dermatome, and you will understand what I'm talking about. Dermatome is the innervation of one particular part of the skin. Okay, it's called cutaneous innervation. It looks like a zebra. Okay, the human looks like a zebra. This part of the skin is innervated by one nerve, this by one, this by other, etc., etc.
We have it in our anatomy book.
You definitely have it. If you open the anatomy book, you've seen it, but some people don't open it. They just do the notes. Okay, so if you open the book, for sure you have seen the dermatome arrangements. Yeah. Okay, so dermatome is the innervation to the skin. Is that clear? Okay, this is also not important. Just want to get your attention. If you look at the embryo, retinoic acid accumulates cranially. FGF8, WNT3 accumulate caudally. This is important for creating somites, because do you see somites, they start in occipital region and they're going to the caudal region, right? So the pairs appear towards the tail. Why? FGF8 attracts them. Okay, so you're going to see that retinoic acid causes the cranial area of the somites, of the paraxial mesoderm somites, while FGF8 the caudal. This picture will come up. Okay, they're going to bring this picture. You can see that this is the neural tube. This is the surface ectoderm, which is your epidermis. This is the notochord. So how do we call this? This is your paraxial mesoderm. But how do we call one segment of paraxial mesoderm? Somite, very good. So this is one somite. Another somite will be right next to it. From head to tail, how many somites are you expecting initially? 42 to 44, right? So that's how many somites there would be. What does each somite consist of?
Myotome, dermatome, sclerotome.
And sclerotome. Agree with me that sclerotome should be closer to the neural tube and notochord, because it will give rise to cartilage. These cartilages will fuse around the neural tube and notochord, creating the vertebral column, the vertebrae, individual vertebrae. Dermatome should be closer to the epidermis, because together they will be the skin. And myotomes are always in the brown color, I don't know, yellow, brown color, and they are just in the middle. So either this picture or this picture or this picture will come, and they will point like here, and they'll ask you, what is that? Dermatome is always closer to the skin. Sclerotome is always closer to the neural tube and notochord. Whatever is in the middle is the myotome. Together, all of this is known as the myotome, somite. Last thing, okay, bear with me. This is the last thing, the endoderm. What does endoderm give you? Organs, because it's the inner part of your embryo. Majority of the organs will come from there. But for your self-control test, GI, gastrointestinal tract, is going to be arising from the endoderm. Okay, you cannot talk about endoderm without first folding the embryo. How does your embryo look like right now? For now, it looks like a disc. Do you see? So it has the head, it has the tail. This is sagittal section. This is the cross section of the embryo. How do we call this?
Visceral.
Nope.
Parietal.
Parietal mesoderm, because it is under the ectoderm. How do we call this?
Visceral.
That's the visceral, because it is above the endoderm. This is the amniotic cavity. This is the yolk sac, neural tube, and notochord. I forgot, guys, sorry. I saw a question, dorsal aorta. It's such an unfair question to ask because you have no idea what is dorsal aorta. But about 2 years ago in past papers, I saw that's what they asked. They pointed to this structure and asked you, what is that? It's a dorsal aorta. Embryo has 2 of them. Embryo has 2 aortas. One of them will eventually disappear, but it's not important. Just memorize that that's dorsal aorta, yeah? They asked this 2 years ago. Okay, so we're talking about folding the embryo. And I will try my best to show it on myself first, so you can understand what am I talking about. Okay, I am your embryo right here. This is the head. Somewhere there is the tail. Okay, head, tail, right here. What is in the center?
Notochord.
Notochord. And what is above the notochord? Somewhere there at the back.
Neural tube.
Neural tube, perfect. There is a notochord, and right behind it, somewhere there, there is a neural tube. Right next to my notochord, I have somites, perfect. Paraxial mesoderm or somites. Right next to it,
Intermediate.
intermediate. And here, like that, I have
Lateral.
lateral plate. And this lateral plate is everywhere like that. Everywhere, right? So everywhere is this way. You can see that this is somewhere my face is. So could you remind me which cavity would I have here? Yolk sac. And which cavity would I have at the back?
Amniotic.
Amniotic cavity. We're trying to understand how does the embryo fold. Because if you look at any picture, this or this, your embryo doesn't have a body cavity yet. It's just a disc. It's a flat disc with the head and with the tail. That's it. To fold an embryo, we first elongate the neural tube. Do you see that this is the head and this is the tail? Elongation of the neural tube forces your embryo to assume this position. This is the head, this is the back, and this is the S. How do we call this position?
Fetal position.
Fetal position. Embryo started with a straight back, but because of the elongation of the neural tube, embryo now looks like this, okay? Embryo is now fully arched, right? So it was straight, but now it is fully arched like this. Now, can you imagine if I am elongating my neural tube at the back, I'm constantly folding a little bit like this. Which cavity you said is here?
Yolk sac.
Yolk sac. I'm constantly folding. What am I doing to my yolk sac? I'm kind of pushing it towards the middle. Am I right? I was straight, but now I do this. I am definitely pushing my yolk sac to the middle. So do you see? Eventually, when the embryo is done assuming fetal position, the yolk sac is going to be in the middle only. You will not see it in the cephalic region, nor would you see it in the caudal. It will be only in the middle. I repeat, you started with yolk sac being everywhere, head to the tail. But because of the cephalocaudal flexion, you see, your cephalic region have flexed, your caudal region have flexed. So because of the cephalocaudal flexion, your yolk sac ends up in the middle only. So now, if I show it to you on the paper, your embryo, I repeat, head, tail, your embryo was straight. Now your embryo looks like this, right? The back is arched. Is it clear? I shift your embryo towards you. This is the head. I shift it towards you. Could you tell me what are these?
The lateral.
Amazing. These are the lateral plate. Is your embryo folded? Not really, it's not folded. It's simply curved, but it's not folded. So what should the lateral plates do to fold the embryo?
Fold toward the middle.
They should fold as well. So something like this should be happening. Your lateral plate should come close together. Do you see? That's the head. Your lateral plate should be approaching one another. And this is what you can see on this picture. Do you see? That's your neural tube. That's your two lateral plate regions. Remind me, how do we call this?
Somato.
Somato what? Pleur. Somatopleur. Do you see that two somatopleurs are approaching one another? So they can fuse together. And as they fuse together, a body cavity arises. But just a second. Here I have my two somatopleurs. They're coming close together to fuse. Is there anything on their way here?
Yolk sac.
But what we just said, where is the yolk sac? Is it here? It's in the middle, but it's not here. Do you remember you pushed your yolk sac to the middle? So if the two somatopleurs are trying to fuse in this region, in the cephalic region, is anything stopping them?
No.
Guys, yolk sac is in the middle. Your two somatopleurs are trying to fuse in this region. Is anything stopping them from fusing?
No.
Nothing. If the two somatopleurs are trying to fuse in the caudal region, is anything stopping them?
No.
No. But if two somatopleurs are trying to fuse in the middle, can they?
No.
No, they cannot, because there is a yolk sac. You have two pictures here, C and B. If I do a cross section through here, cross section like this, will it look like C or B?
C.
C. If I do a cross section through my tail below, will it look like C or B?
C.
C. But if I do a cross section through the middle, will it look like C or B?
B.
B. The two somatopleurs cannot fuse in the middle, but they can fuse in the cephalic region and in the caudal region without a problem. When the two somatopleurs are fusing in the cephalic and caudal region, they are forcing the endoderm to become a tube there. This tube is called gut tube. But if we draw this gut tube, this is how it will look like. It will have three regions. In the cephalic region, it is a tube. Do you see this endodermal tube? It is a tube. It's this. It's a tube. We call it foregut. This is called foregut. In the caudal region, where we said it's a tail, if we do a cross section, it will look like that as well. It's called hindgut. But do you see that in the middle, midgut, it's not a tube. Do you see? It has a connection to something. It will look like this. This tube is not a tube. It has a connection to something. To what?
The yolk sac.
To the yolk sac. Midgut is the only region that has a connection to the yolk sac. And that connection is called vitelline duct. I'm going to call it vitelline duct. So do you see that here is the yellow component? Yellow is the endoderm. Do you see that in the cephalic region, it's a tube. It has two sides to it. It's a tube. Do you see it? In the caudal, it is also a tube. It has 2 yellow compartments. But in the middle, do you see it's not a tube? It's like it's continuing to the yolk sac. So due to the folding of the embryo, when you did this, I repeat, these are your 2 somatopleurs. They did something like this. They have come close together, right? They folded together. My hand is what?
Yolk
The yolk
sac.
sac.
Yolk sac. So if you do a cross section through the cephalic region, do you see it's a tube? It's fully closed. We call it foregut, the endodermal foregut. If you do it through the tail, it's also a tube. It's no problem. It's the hindgut. But through the midgut region, what am I also connected to? I'm not separated, right? I'm connected to the
Yolk sac.
yolk sac. So how would my fingers be called now?
Vitamin.
The
Fine duct. Is that clear? Will yolk sac be forever? When does the yolk sac disappear? I told you in our first lesson. Yolk sac disappears during the 3rd month. Vitelline duct will disappear 3.5 months. Eventually, your midgut will also be a tube, like you right now. Okay, you're a cylinder. Your body is a cylinder. That's the head, that's the tail. You're a cylinder right now. So is it more or less clear what happens to endoderm, right? You can't talk about endoderm, what does it do, unless you're talking about the folding of the embryo. So I repeat that the first folding of the embryo is actually cephalocaudal flexion. Your embryo simply does this. It has an arched back, that's it. But the second folding is called transverse folding, when the 2 somatopleurs from these sides, they come close together. And as they come close together, they push your endoderm into a tube. This endodermal tube is called gut tube, and gut tube has 3 regions. Closer to the head, this is your foregut. Closer to the tail, this is your midgut, hindgut. But in the middle, this is called midgut. And technically, it's not a tube because something was preventing the closure of the 2 somatopleurs. And that something is yolk sac. So midgut is the only one that has connection to the yolk sac. Foregut does not. Hindgut does not. I repeat, this is the foregut right here. Do you see any connection to the yolk sac? No. This is the hindgut. The only connection it has is to allantois, but we'll talk about it next semester. It's not important this semester. So midgut is the only one that has connection to the yolk sac, and that connection is called vitelline duct. I know that it's very confusing, so I will send you additional video of a guy doing it with like Play-Doh. It's actually quite understandable there. Okay, so Play-Doh, he shows how the thing folds. So you will try, so you will be able to actually see, because for sure, I think you don't get it entirely with the paper and me. But is it more or less clear? We put the yolk sac into the middle due to the fetal position folding. 2 somatopleurs are folding everywhere along my embryo. The problem is that in the middle, they can't because there's yolk sac. Yeah? No? It's a definite no. I can see it in your face. It's a definite no.
I have a question.
Yeah.
So the pleurs are basically moving in because of the cephalocaudal flexion?
That's a very good question. In a way. So both the cephalocaudal flexion and the somatopleurs coming close together are controlled by one thing. And that one thing is the lengthening of the neural tube. The neural tube starts to elongate, yeah? And as it elongates, it forces your embryo, which is disc, to kind of start folding into a cylinder. Yeah. It can't, the neural tube, as it elongates, it can't go straight forever. It can't. It will eventually force your embryo to curve into something. Yeah. And that's the curvature. The first curvature is that it has the arched back, which we see on this picture. And once this is done, you're going to see that there's another folding, which is from the two sides. Do you see that it's also because of the neural tube, because somatopliurs have ectoderm in it. So there are lots of growth factors, lots of physical parts that are responsible for that. Yeah. So yeah. Okay, so two last slides, this one and this one. I'm gonna let you go home. This is a blah, blah, blah. Okay, like we'll do it on past papers, blah, 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 honestly. Two important things. An entire pregnancy could be counted depending on when do you start counting. Do you start counting from the last menstruation or after fertilization? If you count from the last menstruation, it's approximately 40 weeks or 280 days. If you count from after fertilization, it's 38 weeks. Could you tell me what is this 2 weeks difference? Why? Why 2 weeks exactly?
Because we're waiting for the, I think this is completely wrong, but we're waiting for the progesterone levels to go down or something like that. No?
It takes 2 weeks for ovulation.
Ovulation, very good. Ovulation happens day 14. So that's the 2 weeks difference. Do I know when exactly fertilization happens? Well, it should be 1 or 2 days after ovulation. Yeah, so that's the only time you can get pregnant. So 1 or 2 days after ovulation. So it's very difficult to count it. That's why we don't. So in medical practice, we only ask last menstruation, which is 2 weeks before your ovulation. So that's where the 2 weeks come in. Okay.
But if you have the semite on the 20th day, isn't it more accurate to follow the pregnancy from? If you have the first pair of semite on the 20th day, isn't it more accurate?
You're confusing. We're not talking about the somites. Somites are for the paraxial mesoderm. We're talking about pregnancy, an embryo as a whole.
Okay.
But you know, it's very costly. It's very, first of all, costly. I can see what you're doing. Just like do the minus 20 days, yeah?
Yeah, just to be more accurate with the following of the pregnancy.
The problem is, you guys may not know this, embryos don't like ultrasound. Every time you do an ultrasound, they turn away from it. Why? Because can you imagine a sound wave going in the amniotic fluid? It actually creates a wave. Can you imagine? I keep throwing waves at you. Would you like that? Not really. So every time you do an ultrasound, embryo keeps turning the back like this. He keeps turning, like you move it and he also turns it with it, you know? So it's very difficult to do that. So that's why it's a very good idea though, but it's not practical. Like you'll have to be professional sonographers to do that for sure. But no. Okay, so pregnancy, we talked about that, the numbers. There are 2 ways to measure the embryo. There is a crown rump length, which is from the crown to the rump. Rump is the old name for ass in British language, okay? Rump, crown rump length. And there is a crown heel length. That's another way to measure it, from the crown to the heel. Another name for crown heel length, it is also known as the standing height, because it's from the crown to the heel. Another name for the crown rump length, it is also known as the sitting height, because it's from the crown to the rump. You need to know that when you're born, you should be about 3.5 kilos, crown rump length 36, crown heel length 50 centimeters. Okay, so yeah. There are 2 very important names that you need to remember. Until the 9th week, the period is known as the organogenesis. So from the moment of fertilization until the embryo is 9 weeks old, you call that period organogenesis. So the name is understandable, right? We're making organs. This is the time when the embryo is most vulnerable to things, okay? So like drugs, alcohol, I don't know, antibiotics, etc. So you can do some damage to embryo. But after the 9th week, this is known as the fetal period. So technically, 10 weeks old human should not be called embryo. You should call it fetus because it already has all of its organs. It has them. The only thing which is left to do is to mature these organs. That's it. So is it clear? Until 9 weeks, you're an embryo, organogenesis. After 9 weeks, you're a fetus. You have all of your organ systems that need maturation. 3rd month of pregnancy, length of the head is half of the crown rump length. Can you imagine when you're 3 months old embryo, your head is until here, half of it, exactly half is the head. I'm sitting right now, half of it is the head. Head is very important. But by the 5th month, head is 1/3 of the crown heel length. Did it shrink? No, your body grew. Your body caught up with the head. Okay, so now you have your normal things. Weight of the embryo during the 5th month is only 500 grams. Fetus is starting to be covered with the fur, yeah, this peachy hair. This fur or peachy fuzzy hair are called lanugo hair. They should disappear when you are born, okay, but sometimes they don't, but within 1 year they will, because you know we're mammals, we should have fur on our part. So that fur is called lanugo hair. 6 months of pregnancy, skin is reddish and wrinkled. Why? Because we still lack connective tissue. All of the blood vessels are very exposed. They're literally under the epidermis, yeah? The connective tissue is lacking. When you're born, you're covered in this whitish substance, this cheesy, fatty, whitish substance, which is called vernix caseosa. Okay, if you have ever seen a live birth, you would have seen that it's this little tiny cheesy thing that covers the embryo or fetus. What is the purpose of fatty substance? To protect the embryo. Can you imagine for 9 months you're in the amniotic fluid? Sometimes you take a bath for too long, already all wrinkled, you know? You wash the dishes, already the hands are wrinkled. What about 9 months in the fluid? Definitely I need to make sure that fluid is not entering or leaving the embryo. That's where the vernix caseosa comes in. Last thing for today, I swear it's just 5 minutes, I'm gonna let you go. Twins. We have 2 types of twins. Monozygotic are on here and dizygotic are on here. Another way, monozygotic are identical twins. They look exactly the same. Dizygotic are non-identical or fraternal twins. Which ones are more common? Of course, dizygotic. Monozygotic is 1 per 1,000. It's quite rare. Okay, so you're gonna see that it's not as common. Dizygotic are about 4, 6, 8 per 1,000. So they're more common. How do dizygotic twins arise? Instead of 1 oocyte, you have ovulated 2. Instead of 1 Graafian follicle, you had 2 Graafian follicle, right? So they ruptured, you have 2 oocytes. 2 spermatozoas are going to come in and fertilize them. So you have 2 zygotes. Are those zygotes identical? No, you have 2 different oocytes fertilized by 2 different spermatozoas. The genetic information is completely different. So it's just 2 siblings. They aren't really twins. They're just siblings that are sharing the womb at the time of birth. Yeah? Of course, they have some kind of similarities, but that's because they're brothers, sisters. They're siblings, but not because they're twins. When they ask you questions about twins, they only want to know what do they share. Dizygotic twins share nothing. You're going to see 2 placentas, 2 amniotic cavities, and 2 chorionic cavities. They share nothing because it's 2 zygotes, right? They share absolutely nothing. Why share if I can make my own? Monozygotic twins arise from one zygote. You had one oocyte fertilized by one spermatozoa. Monozygotic twins arise due to splitting of that zygote at different stages. The most common when it can split is the 2-cell stage. Remember, zygote, cleavage, it becomes 2-cell stage. Randomly, nobody knows why, these 2 cells split, they separate from one each other. Now I have one cell, and they start to divide and turn into monozygotic twins. They come from one zygote, right? So you had one zygote, gave you 2 cells. These 2 cells split, they separated, gave you 2 human beings that look exactly the same, share the same identical genetic information. Do they share anything, the ones that have split at 2-cell stage? No, because it's so early, right? 2-cell zygote. Why share if I can make my own? You're going to see 2 placentas, 2 amniotic cavities, and 2 chorionic cavities, the same as here. Now, the second stage when they can split is early blastocyst stage. Instead of one inner cell mass, for some reason, they make 2. You have a blastocyst with 2 inner cell masses in it. For sure, you have to split, because another name for inner cell mass is the embryoblast or embryo proper. This is a human being right here. So for sure, it has to split, and it does. The problem that it's a bit late, so they will share. The twins that separate at blastocyst stage, they will share placenta and chorionic cavity, but they will have 2 amniotic cavities. They'll have different amniotic cavities. The rarest one, the most rare, the one that barely arises, is that you start separation at gastrulation stage. What is more important, splitting or gastrulation? What's more important? Gastrulation. Making trilaminar germ disc is more important. Just a second, guys, sorry. Literally, I have like one sentence to say and the delivery comes in, of course. The gastrulation stage is more important. Guys, sorry, just a sec. I'm so sorry, guys. Sorry. In Poland, you know, everything is late. Everything. They're so late every time for everything. Anyway, we're talking about the last thing here. So just 2 last sentences. The monozygotic twins that separate at the gastrulation stage barely separate. And for sure you have seen them vis-a-vis Siamese twins, yeah? The conjoined twins, the one that share some kind of body parts. So they separate at the gastrulation stage. Is it impossible to get healthy babies out of the stage? No, okay, it's not impossible. You can deliver healthy babies that are not conjoined, but it's rare because this stage of separation is very late. Okay, gastrulation is more important, so you don't finish the splitting itself. So in this stage, you're going to see that they share everything. Placenta, amniotic, and coronary cavities, every single thing is shared. So quick recap, monozygotic twins arise from one zygote, one cell. This cell can split into 2 at different stages. The earliest one, 2-cell stage. Middle, blastocyst stage. Instead of 1 inner cell mass, you have 2. Latest, gastrulation. Is it clear what do they share and what do they not? Okay, thank you so much, guys. Sorry I made you wait. Okay, so that's it. We have finished. So yeah, we have finished. So that's all the embryo.